In this video, we will demonstrate the power of a modified Coomassie staining protocol for detecting several nanograms of protein in polyacrylamide gels using the example of two-dimensional gel electrophoresis. Prior to first dimension separation, immobilized pH gradient strips are rehydrated in denaturing buffer conditions. After overnight reswelling of the strips, protein samples are applied via anodic cup loading. During the focusing process, the proteins are separated according to their isoelectric points. Second dimension separation is performed by SDS-gels. The gel is then transferred to a staining dish and the proteins are detected by staining with colloidal Coomassie.
Hello, I am Nadine Dyballa from the laboratory of Sabine Metzger in the Biological Medical Research Center at the University of Düsseldorf. Today I will draw your attention to a widely non-famous procedure according to Kang and colleagues. Coomassie Brilliant Blue is a dye commonly used for the visualization of proteins after electrophoretic separations, but is often assumed to be less applicable for comparative proteomics. However, Kang's colloidal staining offers superior features concerning sensitivity, cost and practicality. We use Kang's staining protocol in our laboratory for nearly every gel-based application, but especially in analytical proteomics to determine myocardial protein expression changes. I will demonstrate the use of this method to detect myocardial proteins in the alkaline pH range of two-dimensional electrophoresis gels. So let's get started!